In the labyrinthine process of drug development, analytical procedures stand as the unsung heroes. They are the unsparing gatekeepers, meticulously validating the safety and efficacy of every new pharmaceutical solution that emerges from the crucible of scientific innovation. Analytical procedures, at their core, are methods employed to scrutinize a drug's substance. They are validated to ensure their reliability, accuracy, and suitability, acting as a litmus test for the drug's performance, but they don't stop there. They also gauge the appropriateness of the methods for detecting and quantifying impurities. The analytical procedure's precision is further fine-tuned by setting a quantitation limit. This is the lowest level of an impurity that can be reliably measured. It's a critical safety net, ensuring that impurity results are reported if they exceed this established threshold. Organic impurities, those pesky unwanted chemicals, are measured through various techniques, such as comparing analytical responses. Reference standards, substances known for their specific reactions, are used as benchmarks in the analysis. When it comes to reporting the impurity content, the devil is in the details. Results for all batches are crucial, be it for clinical, safety, or stability testing. And it's not just about passing or failing. Precision matters. Quantitative reporting, using numerical values, is preferred over broad terms like complies or meets limit. Now, science is ever evolving, and so are analytical procedures. When changes occur, it's crucial to link reported results to the specific procedure used. New procedures require fresh validation, ensuring the continued integrity of the process. Batch information, too, holds significant weight. Details like identity, size, date of manufacture, site, and manufacturing process provide essential context for the reported results. It's the backstory that helps us understand the journey of each drug substance. Moving on to the specifications, these outline the criteria that the drug substance must meet, including allowable impurity levels. The decision to include or exclude impurities in the specification is based on observed impurity profiles in safety and clinical development batches, as well as those from the proposed commercial process. The acceptance criteria, those predetermined standards that impurities must meet, should be based on safety data and the capabilities of the manufacturing process and analytical methods. In a nutshell, analytical procedures are the rigorous checkpoints in the marathon of drug development. They ensure that every new drug that hits the market is safe, effective, and suitable for its